Hi, everyone. Thanks for joining us for this virtual panel discussion for Cell Press Cancer Research Week. I'm Krista Bledsoe, and I'm an editor at Molecular Cell, and I'll be moderating today's discussion on tumor metabolism. Tumor cells exhibit metabolic alterations that support their increased proliferation, and they can often acquire nutrients from the extracellular environment to support their cellular metabolism. So today we're going to discuss the links between diet, extracellular nutrient availability, and cellular tumor metabolism. And so joining me today are three experts in the field of tumor metabolism. We have Jason Locasale from Duke University, Katy Wellen from the University of Pennsylvania, and Thales Papagiannakopoulos from New York University. So let's get started. The first question I want to ask is, how do tumor cells sense and take up nutrients from the microenvironment? And what is the contribution of extracellular uptake compared with de novo synthesis? So I'll let everyone weigh in here. It's a broad question, but we can start with Katy if you want to give us some thoughts you have on that.
Okay, sure. So I guess first of all, acknowledging that this is a very broad question that we could probably devote, an entire meeting to. But I guess we can maybe break it down into the three parts of this question. The first being, how do cells sense nutrients, and then what determines how they take up nutrients from the environment, and then third, sort of that contribution between de novo synthesis versus uptake from the environment. I guess in terms of how cells sense nutrients, if we can maybe start there. Of course, there are your core nutrient signaling pathways that are critical for this. That would include AMPK, that responds to cellular energy charge. When nutrient levels are low in the cell, it mediates different adaptations to allow cells to shut down energy consuming pathways and activate pathways for energy production, for example, increasing fatty acid oxidation, increasing autophagy, and so on. Likewise, the mTOR signaling pathway is also, of course, really critical for nutrient sensing, responding to availability of amino acids, among other inputs, and playing a key role in promoting growth and tumor metabolism. There are other ways that cells are responding to nutrient availability, and one aspect that my lab has been really interested in, and Jason's lab has been really interested in as well, is the notion of metabolites playing key roles as signaling molecules by virtue of the fact that they can be used as substrates for protein and chromatin modification. So our lab has done a lot of work, particularly in the area of acetyl-CoA metabolism, which has pretty striking effects on acetylation of histones, for example. Jason can maybe speak about his work on that as well, and methionine metabolism potentially. Those are some of the broad areas, some of the broad ways that cells can sense and respond to nutrient availability and metabolite levels within the cell. I can talk, maybe I'll turn it over to another speaker to address some of the other aspects.
Sure. Jason, do you want to jump in next, or?
Sure. Yeah, I think Katy mentioned a lot of really important aspects of basic mechanisms of how cells sense nutrient availability. Just to add on to some of those, and particularly with the work that Thales has been doing the last 5-10 years, is there's also a lot of the metabolism that occurs in cells leads to differences in redox states, and there's just as a result of doing all the chemistry involving moving electrons around in different metabolic processes, cells need to figure out a way to communicate the status of that redox availability, and that's something that Thales's lab has done a lot of nice studies, and a lot of others as well has done a lot of work figuring out some of those basic mechanisms and how they're aberrantly regulated in cancer. Like there's a really interesting interaction with this protein KEAP1 and this transcription factor NRF2 that upregulates all kinds of things to adapt to differences in redox metabolism and metabolism, and given that redox metabolism is coupled to everything else in metabolism, there's all kinds of interconnected pathways. Something that we've been really interested in, I guess since I started my lab, or even before that when I was a postdoc, was how certain essential amino acids and how certain essential amino acids get metabolized, where they come from, and then figuring out, and that got us into a lot of thinking about nutrition and diet, because these amino acids, like this amino acid methionine, essentially we showed that just changes in diet and physiological context could lead to complete reprogramming of the pathway, like in very acute amounts of time, just coming from changes and differences in diet, and that was propagating all the way to cellular changes, and had all kinds of influences on things like redox, things like chromatin modifications, and so on. So there's just all kinds of different pieces of machinery that exist that cells use to adapt to the environment that involves their nutrient availability.
Mm-hmm. Thanks. Thales, do you want to weigh in on that question a little bit?
Yeah, so I mean, I think both Katie and Jason already gave some really nice background about some of the sensing mechanisms. So as Jason mentioned, we've been particularly interested in understanding how redox stress and how oxidative stress is sensed by cells, and the pathways that do this, and how they reprogram cellular metabolism to allow us to adapt to redox stress and cope with it. And the same pathway actually is interestingly also responds to certain electrophilic metabolites, so certain metabolites such as fumarate or methylglyoxal, or for example, that can be derived from glycolysis, or other electrophilic metabolites, which, because of cellular rewiring that occurs in cancer cells, may accumulate. And this NRF2 pathway is a way by which cells can respond to clearing some of these toxic electrophilic metabolites. So the work from our group, and also Gina DeNicola's group, has done put a lot of effort in looking at this. Another sensor mechanism, I guess, that I think is very important, particularly for amino acids, is the ATF4 pathway, which is essentially responding to lack of amino acids for translation, which is all very important step. Obviously, if you don't have amino acids to do translation, the cells respond by activating ATF4, which essentially turns on this whole program of factors that can both generate amino acids, but also uptake them from the microenvironment. And I guess two other things I want to bring up is sort of the role of the importance of, so not just antioxidants and generating them or uptaking them from the environment, but also like things like vitamins or iron. So, which are very important for producing a lot of the cellular metabolites. Iron, for example, is also involved in regulating, for example, epigenetic reactions, such as the deoxygenase enzymes, right? So, which can demethylate DNA and histones. So, so regulating iron levels is critical, and that can happen at many different levels. For example, NRF2 does this. It's sequestering iron, but there's also very fundamental push transcriptional mechanisms that regulate some of the iron transporters. And for example, Richard Posamato here at NYU has done quite interesting work on this, looking at iron regulation in cancer cells. Yeah, I think ultimately, in terms of like the how cells manage the uptake versus synthesis, I guess there's like a balance, obviously, and it really depends on like the availability of nutrients. But ultimately, I guess I would think that potentially, if nutrients are quite available, they would preferentially uptake them versus like generate them themselves, which requires a lot of like important cofactors to generate reducing molecules, and it requires NAD, NADH, NADPH, for example. So, I think when you look at the balance, I think uptake probably would be preferred. Obviously, it depends on like what kind of environment the cancer cells are in, so they have to adapt. And I guess overall, a lot of elegant work from Katy and Jason, many others have shown that, like, cancer cells are very plastic, so they can switch quite easily from uptake to synthesis, right? And I guess we'll discuss this later, but this is also an issue when trying to target metabolism, right? How interconnected all of these sensing pathways and uptake pathways from the environment or synthesis pathways are. So blocking one essentially triggers another, and the cells can really adapt very rapidly.
Interesting, thanks. Appreciate those are some interesting insights. We can move on to our next question, which is, how does diet affect the availability of nutrients in the tumor microenvironment and lead to alterations in tumor cell metabolism at the molecular level? And I think Jason mentioned this a little bit already, so maybe we can start with you about how diet affects cellular tumor metabolism.
Oh, okay. Yeah, it's another great question. Another question where we could write 50 pages and have several multiple day meetings on. But I guess the basic idea is that all metabolism in cells, there's an input, there's a source of like any kind of biochemical reaction that involves that involves chemistry, requires substrates and requires origins of where those. So the substrates have to come from somewhere, and ultimately that's what life is that nutrients from the environment get chemically modified in ways to undergo ways to support life in the form of things like energy, signaling functions, and biosynthesis and so on, and the source, the original source of and whether we're talking about mammals, whether we're talking about plants, I mean, there's ultimately they need carbon, nitrogen sources, and those come from the diet. I mean, the original source is the diet. So that being said, I mean, mammals have evolved a lot of complicated things that go from the point at which you have that environmental factor, which is the diet, to the cell biology that we're talking about, and there's all kinds of processes. I mean, just basic concepts of that you eat something, your gut like your gut processes it, your liver processes it, your muscle and all, the whole physiology interacts with it, and eventually what it leads to is defined concentrations of nutrients in your plasma, and those nutrients, some of them are determined, some of them are highly regulated by the liver, some of them are highly regulated by muscle uptake, some of them are highly regulated by what the gut. I mean, Katy did some nice work on fructose in the gut recently, and so but some of these things are not as regulated, and that was some of these things where they're not as regulated through all the physiology, and one of the examples we've been working on a lot in our lab is these essential amino acids, particularly this methionine, where basic changes in methionine content in the diet are propagating all the way through all this complicated physiology to reach to changes in methionine in the plasma. And then once you have these changes in methionine in the plasma, those changes then get uptaken into cells, and then you get all the metabolic reactions, all the metabolic pathways, and all the machinery that's downstream of these metabolic pathways are influenced by it. And so those are and we're just beginning to understand the extent that diet actually changes metabolites in the serum and the plasma, and to the extent that those changes change the availability and change the the wiring and the structure and the function of metabolic networks, and that's it's a major emerging area at the moment.
Thanks. Katy, do you want to weigh in on that as well?
Sure, yeah. So I guess to kind of build on what Jason was saying, I think when we think about how diet is impacting cancer cell metabolism, it's important to think about, as Jason was saying, both the direct effects of that nutrient. He gave the example of methionine intermediates, and how that impacts in a cancer cell, but also the effects more broadly on systemic metabolism. So, for example, we could take branched chain amino acids as an example, right? If you added more or took away some branched chain amino acids from the diet, that would both have effects on impacting the availability of branched chain amino acids to tumor cells, as well as to other cells existing within that tumor microenvironment, fibroblasts, immune cells, etc. And it would have really important broader systemic effects. For example, particularly in the context of an obesogenic diet, high branched chain amino acid levels will contribute to insulin resistance and hyperinsulinemia. And so those secondary effects of hyperinsulinemia can then have very profound effects on tumor cell metabolism as well. And insulin is an example. There are other effects, on adiposity, on inflammatory cytokine production, and so on, that could also be impinging on cancer cell metabolism. So I think we need to, when you do a dietary manipulation, be thinking about both the direct effects of the nutrients themselves, but also the broader systemic effects as well. And then the other point that I wanted to make is that I would say that we still actually have a pretty poor understanding of what nutrient availability within the tumor microenvironment actually looks like. There's been some nice work recently from Alex Muir and Matt Vanderheiden, where they developed methods to isolate the tumor interstitial fluid and measure metabolites there. And they're reporting pretty different metabolite availability in that tumor interstitial fluid from in serum. And so, how diet impacts what's really in the tumor microenvironment, I think, we're just beginning to be able to answer those questions.
Thanks. Yeah, a lot to learn there still. Thales, do you have comments on this?
Yes, I think everything that's already Jason and Kay mentioned is covered quite a lot, actually, of what I want to discuss too. So I think overall, there's a lot of really exciting ongoing work looking at how altering protein, fat, or carbohydrate content in the diet can interact with systemic metabolism, liver, muscle, pancreas, and of course the microbiota. Again, Katie had a beautiful paper recently on high fructose diet and how that impacts how microbiota play a role there in processing fructose and systemic availability of certain metabolites in response to that. And then that will impact, as already mentioned, the availability and of these nutrients in the circulation that are derived from the diet. However, ultimately, again, as Katie mentioned as well, the tumor microenvironment is a very complex environment. First of all, the tissue where the tumor lies is critical, right? So if the tumor is in the liver as opposed to the pancreas or the lung, and potentially tumors even with the same mutations in different tissues may have very different microenvironment and demands for metabolites and utilization of those metabolites. And Matt Vanderheiden's group has done some great work on this topic. And of course, the local, in addition to the tissue itself, where the tumor lies, the other important factors, of course, the content of the tumor microenvironment, right? The combination of the stromal cells, especially the immune cells, the heterogeneity of the tumor itself. So that will, oxygen concentrations, and vascularization of that tissue will really impact sort of the local availability of the nutrients as opposed to what's found local in the circulation. And I think Katie mentioned the great work by Alex Muir and Matt Vanderheiden looking at the tumor interstitial fluid. Another approach I think that's critical, is very important, and it's going to lead to very interesting observations in the future. Has technical limitations though, is imaging mass spec. So really be able to image tumor metabolites in the tumors and in the tumor microenvironment and look at their levels, potentially combine that with tracer methods to really understand kind of where these metabolites are derived from, etc. And another point I want to make in general is that we don't often think about is the temporal aspect of diet and when the diet is actually administered. So there's a lot of work, emerging work in the circadian rhythm field showing that if diets given at different times of day can really dramatically impact the effect of that diet in systemic metabolism and physiology, because the liver and the pancreas and the colon and other tissues are regulated in a circadian fashion to break down metabolites, utilize metabolites, and modulate systemic metabolism. So I think it's, for example, intermittent fasting, right, which is very important. So it's a diet a lot of people are even myself are actually using right now, and that really relates to particular compartmentalizing when the diet is uptaking, and that can really impact how it gets processed and the nutrients that are available in the circulation. And I think this has caught a lot of attention, and the NIH even has a provocative question that is related to, for example, specifically intermittent fasting and how that impacts tumorigenesis. So again, the timing, I think, really matters, understanding the importance of circadian rhythms and regulating certain metabolic pathways also needs to be taken into account. Adds another layer of complexity, of course, to our studies, but I think it's very important.
Thanks. I guess we can move on to our next question, which is how changes in nutrient availability or fuel choice might create therapeutic vulnerabilities that you could target in tumor cells. Maybe you just finished talking there. Maybe we can start with you on this one, please.
Yeah, so I mean, of course, this is a very important point, and both Jason and Katy's labs are investigating this as well. I think ultimately there's different ways we can target tumor metabolism. There's small molecules. So we've been working a lot on some of the small molecules, particularly glutaminase inhibitors. Glutaminase is a very important enzyme that that converts glutamine to glutamate, and their glutamine can be used for various pathways, including fuel as a carbon source for the TCA cycle, as a nitrogen source for synthesis of most non-essential amino acids, and also as a source, contributes to the glutathione synthesis, a very important antioxidant in all cells. So there's been a lot of work on understanding glutamine utilization and breakdown in cancer cells. However, even though there's really good small molecules out there, there's been very little success in the clinic in demonstrating that these molecules, such as glutaminase or other, I would say, metabolic strategies, metabolic inhibitors, in having efficacy in patients. So we think in our lab and others have been looking at whether there's subsets of certain tumor types. We're specifically focusing on lung cancer and looking at whether genetic subtypes of lung cancer are more susceptible to glutaminase inhibition as opposed to others, and whether stratifying patients based on a metabolic mutation can potentially make them more susceptible to a therapeutic strategy. Of course, there's other than small molecules, there's enzymatic ways you can intervene. So using enzymes that deplete metabolites, that target certain metabolites, such as asparaginase, which has been used for, 40, 50 years now in the treatment of childhood leukemia. And of course, dietary interventions. And I think Katy and Jason can speak more to that. So I guess the challenge I see in terms of use, as I mentioned before, is a lot of the data that comes out, that I've seen coming out from using metabolic therapies, particularly for solid tumors, has been you often see stasis and you rarely see regression in the preclinical models. And when you see something like that in the preclinical models, it often means that the therapy either is going to have almost no effect in patients, I think, or minimal effect. So I think that's an issue we need to think about. And this is, again, goes back to the fact that, as we mentioned earlier, there's a lot of plasticity in tumors. And yes, you can target glutaminase or you can target certain amino acids in the circulation, either by diet or enzymes, but often tumor cells will find a way around to getting these metabolites that they need, amino acids or nucleotides or whatever it is. And we may have to actually, in order to go further than just stasis, we may really have to find very particular situations where certain tumor types, either based on their location or their genetics, are very susceptible to some of these therapies in order to be able to get a response. There's not going to be one therapy that works for all cancers, metabolic therapy.
Thanks. Katie, do you want to weigh in on that question?
Sure. Yeah. I think those are really important points that Thales was making. I guess in terms of how I think about this question, I guess I would maybe frame it in two like kind of different ways. One being two distinct ways. One being, what vulnerabilities might arise in a tumor that arises in a particular metabolic context. So for example, we know that obesity enhances the risk of many different types of cancer. And so there may be unique vulnerabilities of tumors that arise in an obese person versus a normal body weight person, for example. And there are examples looking at this in the literature, oftentimes looking at, say using a high-fat diet, for example, to model this in mice. And one example that comes to mind of a study like that is one that Nada Kalani's lab did a couple of years ago, where they had modeled obesity-linked pancreatic cancer in mice and found that the obesity in the obese animals with pancreatic cancer, they had upregulation of arginase 2 involved in the urea cycle, and this created a unique vulnerability only in that subset of obese mice. But I guess then on sort of the other side of things is what I think is a very exciting concept, which is how can you leverage diet to enhance therapeutic efficacy? And we've seen some really, I think, beautiful examples of this in recent literature. One that I think has gotten a lot of attention is Luke Cantley's work on use of a ketogenic diet to enhance the efficacy of PI3 kinase inhibitors by preventing the hyperinsulinemia that would ensue with PI3 kinase inhibition. I think that work has moved on to clinical trials. Another, I think, really, really nice example of that type of work is, and this one coming at it a little bit more from an unbiased screening approach, is work that David Sabatini and Nama Konarak did, where they used CRISPR screening to look at determinants of methotrexate sensitivity and identified that histidine catabolism enzyme was really a major determinant of this, because histidine catabolism actually will deplete tetrahydrofolate in the cell. And so they actually proposed that you could supplement histidine in order to improve methotrexate sensitivity. So I think that type of thing is really exciting. How could we use diet to enhance efficacy of existing therapeutics?
Mm-hmm. Interesting. Jason, do you want to weigh in on this point too?
I think Thales and Katie both mentioned a lot of interesting areas, and just trying to think about ways in which they might connect to each other. I think one of the points that I got from some of Thales's commentary is this general issue of metabolic plasticity and context dependence that's often poorly understood and sometimes even poorly defined in tumors. And like Thales mentioned, there isn't a single drug that that works for all cancer types, and every cancer has all kinds of is in a complex situation involving both its genetics and its environment, but the tissue of residence and everything else involving the environment, like the systemic factors. And so with those kinds of limitations at hand in terms of targeting metabolism, I mean that brings this idea of finding those, defining those interactions between how those drugs work, where those drugs are resistant, and then do the resistant mechanisms can they be influenced by physiological factors and nutritional status and diet? And just like in the example Thales was saying about glutamine metabolism, you've got, okay, so this particular tumor, it's resistant to a glutaminase inhibitor because there's all kinds of other pathways, and then I think where the science is going is figuring out what can actually, like whether say there's 4 other ways in which you could influence the citric acid cycle. If you block glutaminase and say, maybe a diet could influence 3 of those ways. And these are some of the ideas that are kind of emerging and related to some of those studies that Katy was talking about, where you have these interactions between targeting a metabolic pathway and then the environmental factors that affect all the other pathways that are related to it that contribute to the overall either maintenance or proliferation of the tumor. So yeah, we've been doing some of this related to methionine metabolism, where you could show that certain chemotherapy resistant situations involving like, for instance, radiation resistance, you can basically show that if you manipulate dietary influences that affect nucleotide and redox metabolism, that also affects the that changes radiation sensitivity in some tumors. And other examples where like the one like some of the ones that Katy mentioned, where there's certain obesity contexts, or certain aspects of amino acid nucleotide metabolism where the particular pharmacological agent is closely related in how it functions in terms of targeting metabolic pathway and how all the dietary factors around it. And it's surprising that this works at all, because something like diet or obesity status, as there's so many things involving how you actually go from that status to some change in some susceptibility in a tumor cell that's residing in a tissue, whether it's because ultimately it's got to propagate to the plasma, it's got to propagate into the microenvironment from the plasma, and then it's got to go and get metabolized within that particular tumor cell and influence the metabolic pathway. So how the progression of diet all the way into the cellular metabolism that is affected by different therapies is it's still very much a emerging area.
Maybe this leads a little into our next question. Can you talk about the models that you use to study the effects of diet and nutrient availability in cellular tumor metabolism, and what are the challenges of translating the findings from these models into patients with cancer? Maybe Katy, do you want to start us off on this one?
Sure. Yeah. So I think those diets could be like broadly characterized into diets that might be tumor promoting, maybe a high fat, high fructose type of diets, or diets that might be more therapeutic or might prevent tumor growth or reduce tumor growth. Things like amino acid depletion, like Jason's been discussing, or fasting, like Thales was suggesting, intermittent fasting, caloric restriction, and so on. And then there are special diets that might be used to synergize with therapeutics. I guess some of the things that we're beginning to think about in the lab, as I think Jason and Thales mentioned, we've done some work recently on fructose metabolism, and we're interested in understanding how that might impact tumorigenesis. I think fructose is most likely because of the way that it's metabolized, primarily in the intestine and then in the liver. It's most likely to impact intestinal cancer, colorectal cancer, and there's evidence recently from Ji-Hee Yoon about use of fructose supplementation to enhance to promote colorectal cancer development, but it may also impact liver cancer, and that's something that we're beginning to look at now. Another aspect that we're really interested in that I mentioned earlier is branched chain amino acids. We're beginning to look at that And I think the effects on systemic metabolism and hyperinsulinemia may be very important there. So those are some of the things that we're beginning to look at in that space.
Thales, do you want to weigh in on this one?
Yeah, absolutely. So I think, yeah, on one hand is the models of like how we impact, the diet, either in the whole animal by, for example, or it changed the diet composition itself, as we discussed and Katie just mentioned, or using certain drugs or enzymes that target certain metabolites in the circulation. And I just wanted to approach it from the side of the cancer modeling and how do we specifically, because I think that's very important. So often, I think cancer is seen as sort of a black box and all tumors are sort of the same. And then all we change is like everything around them. So I think it's important to sort of like how we model cancer to study these interactions, right? So there's been a lot of course, you could do this there's a lot of in vitro studies, the 2D, 3D organoids, using in vivo like media to sort of like be able to understand how you can alter certain metabolites and how tumor cells will respond, which of course is in vitro and there's obvious caveats there. And then on the in vivo side, there's been a lot of work either with transplants of human cell lines or PDXs in immunocompromised hosts, or syngeneic mouse cell lines, again, transplants. And these have certain advantages. They have the advantage in the case of the human cell lines that you're modeling the mutational heterogeneity of a tumor, and in terms of the different sort of tumor cell populations that exist in a tumor, the different clones. However, they have a caveat that they're in the context of an immunocompromised mouse, which is a major issue, especially knowing how the tumor microenvironment plays an important role. The immune microenvironment plays an important role in cancer, and obviously in the context of emerging immunotherapies, it's important to understand how diets impact those immune cells as well. And in general, the transplant models have an issue because you're sort of like bypassing the whole tumor progression of natural tumor progression that happens from a single cell all the way to a tumor, that as it happens in humans, right? So, the other approach is using genetically engineered mouse models, which is mostly what our lab has been doing. So engineering mice to have specific mutations that occur in humans, where you sort of recapitulate sort of the natural evolution history of a tumor in a mouse, and then you can really understand kind of the different energetic demands that cells have will change during tumorigenesis. A single transformed cell or an early neoplasia is not going to have the same microenvironment and nutrient demand as a highly desmoplastic, aggressive, invasive tumor. So these models allow us to understand, kind of the progression of the disease, the evolution, and how diets, how nutrient availability is different, demands are different, metabolism is different, and how the diets can impact this. And also how mutations can impact this, right? Because there's of course mutations that can impact the nutrient uptake, but also nutrient utilization in tumor cells. And we think some of these mutations are highly selective for in some cancer types. For example, in lung, LKB1, also known as STK11, a very important mutation that regulates AMPK that Katie already mentioned, it's involved in nutrient sensing. And KEAP1, these are very frequent mutations. So we think they're selected for because of the fact that they allow some metabolic adaptation. And I guess the other reason to use these models is because you can actually study the interaction with the immune microenvironment. And there's a lot of work being done on this and how diets and metabolite availability can impact the immune cells. Rusty Jones, Erica Pierce, which is Rathmel, Marcia Highgate, they've all done really elegant work looking at this from the context mostly of T cells, but there's also, of course, the innate immune cells that are extremely important. So again, I think I'm just using, I think all these models that I mentioned, both the genetically engineered mouse models, the transplants, and then in various in vitro models have their advantages and disadvantages, and you sort of need I would say, looking at multiple models in order to be able to make get better understanding of how cancer cells utilize nutrients, uptake nutrients, and how different diets can impact their growth at different stages of the disease.
Thanks. Jason, do you want to share your thoughts on models?
Sure. Just yeah, a few things. Yeah, I guess the overall theme that we been seeing on the news the last like few weeks is that there's this consistent message that all models are wrong, and it's just that some are useful, and where that and the point that really drives is that, I mean, models are really for the kind of question that you want to ask. If you want to understand something about the immune compartment in a tumor, like cell culture is not the has limitations, and if you want to learn something about physiology, cell culture, if you want to learn something about how the liver is interacting with the tumor, or how the gut's interacting with the tumor, there's limitations. But with all that being said, I think a case could be made that a lot of what's inspired a lot of this interest in diet, and diet influencing molecular cellular level metabolism has come from observations in cell culture where just simple changes in the media composition of what the cells experience lead to dramatically different effects on how these cells respond to drugs, like just the nature of what their metabolic pathways are doing, and so we've been learning a lot, especially in the cancer metabolism field over the last 10 years, just doing simple things like changing the glucose levels in the media, changing the amino acid levels in the media, and seeing all the different metabolic rewiring that happens. And those kinds of model systems have led to these kinds of thinking about more broad pictures about how that might propagate to something like a mammal eating something differently, and then that leading to different effects, whether it's in a tumor or in a tumor cell, yeah, I mean, ultimately these concepts need to be extended to humans, and the whole nutritional field in general has been largely limited by a lot of lack of like randomized controlled trials, and a lot of the studies, like at least the clinical studies in humans, ultimately, there's just a lot more work to be done, but in general, I mean, like every model's got a purpose, and it's just a matter, and whenever you work on a particular system, you always have to take a step back and try to understand what you're trying to understand from that system, and that generally goes with all the research we do in our lab, where we're generally pretty focused on the kind of measurement techniques and kind of data analysis, and then once we have those tools at hand, then we kind of go and look at we go and look at all the models that are available, and what we can learn with those models, what the challenges are in those models, and also what the positive aspects of them are.
Thanks. Yeah, I'll move on to our last question. I just wanted to ask everyone what you think the most exciting areas of investigation or most interesting recent discoveries are in this area, kind of connecting diet to cellular tumor metabolism. I guess, Katie, do you want to start us off on this one?
Okay, sure. So I guess I think there's so much that we need to learn in this area yet, as we've touched on throughout this whole seminar. I think we have so much to learn about how different diets might impact, what is in the tumor microenvironment, both through systemic effects and through direct nutrient availability effects. I think it's very exciting to think about how a diet could be implemented as part of a therapeutic strategy for particular patients. When we think about diet and cancer, I think one of the big challenges is compliance. So, of course, in the context of, prevention and so on, that may be very hard. Thales is doing intermittent fasting, but, probably not that many people out there are willing to. Here in quarantine, I'm not really willing to, I must say. Anyway, but in the context of a cancer therapeutic strategy, I think people would be really willing to do that, right? If I knew that a drug would work better if combined with a particular dietary intervention, and I was a cancer patient, I would be very willing to make that change, for example. So, you know, I think understanding where those opportunities for combining diet for maximal therapeutic efficacy is a very exciting area. And from my own interest, I think, you know, I think we shouldn't only think about metabolic targets here, but, you know, one thing that we've worked on, Jason's worked on, is this link with the epigenome. And it may be that there, you know, with distinct metabolic vulnerabilities, this may lead to changes in the epigenome as well. And I think we have very little handle on this currently and how diet may influence that. But, you know, there are a lot of epigenetic drugs out there that are being looked at for cancer therapies. And if they could be leveraged, that would be fantastic.
Jason, do you want to share the interesting investigations you think are going on, or recent interesting discoveries?
Sure. I think we're still just at the very, very early stages of having a molecular understanding of generally anything about diet. In the nutritional field, it's still largely in terms of what's taught in dietetics programs and so on, and what's administered, what's generally administered clinically is largely the concept that a calorie is a calorie. Is basically, if you want to change your diet, you change your calorie content. And then there's these more recently emerging areas of changing the macronutrient compositions in diet, like the fat, like relative fat, glucose, protein content, and then also the timing as well, like what I guess Thales hasn't eaten yet today.
Not yet, yeah.
But it's probably going to interact better with his liver, like during. But so those are all like really interesting concepts, and all of them like still need molecular principles around them. But even within each of those, we're still learning that, you know, just even the balance within each of these macronutrient categories is still having a huge effect on metabolism and cellular metabolism. I guess like carbohydrates and fats have always been appreciated, that there's different fats have different have different physiological properties. Something we've been really excited about and basically focusing a lot on in my lab is just figuring out whether protein content in a diet, you know, to what extent is there variability in protein content to the extent that it would actually affect something involving cellular metabolism. You know, we have these laboratory situations where you can drop out an amino acid in a diet, remove the amino acid from cell culture, but interestingly, that's also something that humans are going to, that humans that take on certain diets, they are going to experience a lot of that in their everyday life, and whether that's actually affecting anything involving cellular metabolism is something that's still largely not understood. I mean, just basic protein content that comes from a plant-based versus a meat-based diet is. We'll have all kinds of differences in branch chain and thionine and other nutrients, and whether that's enough to actually impose differences in terms of how like a mitochondria is functioning, or how the redox state of a epithelial cell somewhere within might its metabolism might be set up these are all really exciting areas, and still a lot of work needs to be done.
Yeah, thanks. Elise, what do you think are the exciting areas?
I think what Jason and Katy already mentioned is I'm also very excited about. I think I'm modulating, I guess, the content of some of these diets. For example, Katy mentioned ketogenic diet, right? So there's a lot even to do there still about in terms of course, this can be combined with PI3 kinase inhibitors very nicely by lowering insulin levels in circulation, like Luke Hanley's lab nicely showed. However, certain tumor types may actually grow better in the context of ketogenic diet, assuming they're high consumers of fat. So, and then there also the content of a ketogenic diet, ratio of monounsaturated versus polyunsaturated is very critical, and of course. An important factor that can contribute to all this, in addition, is the content of the microbiome in the intestine. So I'm also very excited about potentially. So in general in the cancer field, immune-based therapies have really like transformed our thinking about how to treat cancer, and have like basically led to cures. However, there's a large percent of patients in tumors that respond really well to immunotherapy, such as lung and melanoma, that do not respond. Of course, there's tumor types that don't respond almost at all. So there's a lot of data emerging that potentially, first of all, the microbiome plays a role in this. So we know that if you eliminate the microbiome in the intestine, and potentially in other tissues too, you completely block the response of checkpoint inhibitors. So there's been several studies on this. So again, how the diet, studies like what Katy has done, showing how the diet can impact the microbiome, and how the microbiome now can influence anti-tumor immune responses, I think is a very exciting field of investigation.
Sorry, so just to briefly, have you, what do you think about these microbiomes in the tumors, like these kinds of studies that have been coming out really recently?
I mean, I think Matt Meyerson's group has done some interesting work on this. I think there's still a lot to do. At the lung, there's been some really cool data in the lung. Actually, a former colleague of mine from my team, Tyler Jackson's lab, had a nice story looking at the lung microbiome, because again, that's a barrier tissue, so there is a decent amount of microbiome. Nothing like the intestine, obviously, but they're there, and they're important for like regulating a lot of innate and adaptive immune cells. So yeah, I guess there's still, I think, a lot to do. I can't really comment one way or another. I think it's really early stages, and definitely nothing's really been done in the context of like metabolic interventions, right? So but again, I think it's very important to sort of understand how altering diet can potentially improve anti-tumor immune responses, right? So and because I think, again, combination therapies, as already was mentioned, is going to be critical. So yes, potentially a metabolic strategy alone might not work very well. Or a dietary intervention might not work very well, and immune-based therapy with checkpoint inhibitors might not work on its own, or not work well enough on its own. But potentially combining these two might give us some sort of synergy. And I think a lot of the work that's been done to understand sort of like how, in the context of the tumor microenvironment, tumor cells and immune cells compete for nutrients or exchange nutrients, is very important. So potentially by understanding how this process happens, we may be able to sort of alter, again, either diets or drugs, small molecules that target metabolism, or enzymes that degrade metabolites, be able to improve immune-based therapies. And yeah, I think that, so I think really like the combination, understanding how we can improve immune system responses is critical. And I think there's very little, I mean, we know quite a bit about T cells, but when you just look at the innate immune cells, which are really also critical for these responses, there's very little understood there about how a lot of the work is in vitro, and we don't really have a lot of good in vivo data understanding how, for example, macrophages or neutrophils, how their metabolism is different in the tumor microenvironment, and how we can impact their differentiation and their function by altering the diets. Yeah.
Thanks. Yeah, with that, I think we've come to the end of our virtual panel. I'd like to thank you all again for sharing your thoughts today. It was really interesting. Jason Locasale, Katy Wellen, and Thales Papagiannakopoulos. Thanks again. And I'd also like to thank you, our audience, for joining us during Cancer Research Week here at Cell Press. And you can head over to cell.com to find our Cancer Research Week page, and you'll find cancer research published across Cell Press journals, and you can access more virtual panels like this one. So thank you again.